There's a lot of information just on this first page, which is a summary of the results. For the bile acids section on the summary page, we have the key information here at the top in terms of the total bile acids that are detected in the stool sample. So the totals themselves really mean a lot. There's a lot of correlations and even mechanistic studies showing that they play a big role in motility, for example, et cetera. So we know that the total amount of bile that gets into the colon is important, particularly for motility. We know high levels tend to promote fast motility. Low levels tend to promote slow motility. Then you can also see here just underneath that, there's the secondary bile acids in terms of percent of all the bile acids, which ones are secondary, which ones are primary. Secondary are the ones that are converted by the microbiome, mostly in the large intestine. So when you have lower than optimal levels, like we see here, that can mean that your microbiome overall is just not very diverse. These are specialized functions to basically make those secondary bile acids. So when you see that deficient, that usually is a good indirect indicator of diversity of your microbiome. The flip side of that is that because you don't have enough secondary, they're not getting converted from primary, you can have an excess of primary. So you'll see a higher percent than optimal. So that gives you your first clue that there's a pretty significant imbalance here. So really a lot of great information just from these first three indicators here that give you a clue as to what's going on, and then may start to help you understand how you might want to approach that from a treatment or support standpoint.